Welcome back to this next video in which we are discussing the visual system. Okay, so in this next video we're now going to turn our attention onto the process of phototransduction, which is the process whereby the photoreceptors, the rod cells and the cone cells, can turn visual stimuli, i.e. photons arriving within their outer segments, into an electrical signal. Okay, so that's what we're going to study, and we're going to start off by studying this process within the rod cells, and then we'll have a look at how it's different in the cone cells. So we'll take it into the more complicated setting of the cone cells. So we'll start with the rod cells, which is very simple because it's only one type of rod cell. That's the hugely complicating factor with the cone cells, that you have 3 different types of cone cells. So we'll save those for later. We'll firstly have a look at this process in detail for the rod cells. So let's begin. So I'll start by drawing a picture then of a rod photoreceptor outer segment here. So remember, it's in the shape of a rod, hence why the cells are called rod cells. And inside this rod, what you will have is lots of these discs of membrane, like so. And this is, remember, to increase the surface area of membrane so that we can put receptors for light inside that membrane. So, inside the membrane of these discs then, what you're going to have is these proteins that are receptive to light, the actual proteins that are going to respond to light, to photons hitting them. They're going to absorb the photon, change state, and then set off intracellular signaling cascades that will lead to an electrical disturbance within this cell, an electrical signal within this cell. So what I'm drawing now is some turquoise blobs in the membrane of these discs, in the membranes of these discs rather, and these turquoise blobs are going to represent these proteins that will respond to light, the light receptors if you like. Okay, so you could continue it on for all of these discs here. So the name of the protein that responds to light inside rod cells then is rhodopsin, and you will most likely have heard of rhodopsin before. It's a very famous receptor. Okay, so rhodopsin is the name for the integral membrane protein that is inside the membrane of these discs here of the rod cells, and which will respond to light. So when photons hit rhodopsin, it's going to change the structure of the rhodopsin receptor, and it will then activate an intracellular signaling cascade that will lead to electrical signals being generated inside this rod cell. Now, rhodopsin is made up of 2 separate components. It's made up of a component called opsin, which is the protein, and it's made up of a small molecule called 11-cis retinal. And we'll begin by discussing opsin. So let me draw the membrane of one of these discs, and to orient you, this side will be the side facing into the center of the disc, so we'll call that the lumen of the disc, and this side will be the cytoplasmic side. Okay, so those are the 2 sides of this membrane. So this is one of the phospholipid bilayers of one of these discs. So now I'm going to draw an opsin protein in this membrane, and the opsin protein is a Gprotein coupled receptor, which means that it has this classical membrane spanning topology where it has 7 membrane spanning alpha helices. So this is representing the polypeptide called opsin, and as I say, opsin is an example of a GPCR, which stands for a Gprotein coupled receptor. So GP is for Gprotein, and then the CR is for coupled receptor. So Gprotein coupled receptor. And Gprotein coupled receptors are a massive, great family of receptors inside humans. We have a huge number of different Gprotein coupled receptors. The famous ones are, for instance, the adrenoreceptors, also the muscarinic acetylcholine receptors, loads of the serotonin receptors, the dopamine receptors. All of these are examples of Gprotein coupled receptors. So Gprotein coupled receptor. So opsin is another example of a Gprotein coupled receptor, and as you can see from this picture, it has these classical 7 membrane spanning alpha helices. So there are 7 there, and all Gprotein coupled receptors have that. Okay, right, so there's opsin. The other component of rhodopsin is a small molecule which binds to this. So this is a protein, this is a polypeptide, this is what this line is representing. It's representing a polymer of amino acids that is spanning the phospholipid membrane in this way. Okay, the other component of rhodopsin is a small molecule by the name of 11-cis retinal. Okay, and for the purposes of this diagram, I'm just going to represent 11-cis retinal as a little blue blob, and it will be bound to the opsin protein, like so. And together, opsin plus the 11-cis retinal makes rhodopsin. So what I've got here is now a rhodopsin receptor, which will be sitting in the membrane of one of these discs inside the rod. Okay, so that's what rhodopsin is. What I want to do now is talk to you a little bit more about 11-cis retinal, because having a good understanding of 11-cis retinal is absolutely essential to understanding the phototransduction process, because it's actually the 11-cis retinal molecule that's going to absorb the photon and change its structure in response to the photon. And when it changes its structure, it causes a change in the structure of the entire opsin protein, and that then leads to the activation of a signaling cascade inside the cytoplasm of the cell. So I want to discuss the structure of 11-cis retinal so that we can see how it's going to actually change its structure in response to absorbing a photon. So I want to actually draw out the structure of 11-cis retinal, and before we do that, I firstly want to show you the structure of Alltrans retinal, and to do that I would like to show you the structure of Alltrans retinol. So we'll start off by discussing the structure of something called Alltrans retinol. Now, Alltrans retinol is actually a very famous molecule. This is usually what people mean when they say vitamin A. Now, there is a little bit of discrepancy there. Some people will use the term vitamin A to mean a huge variety of associated molecules to Alltrans retinol, but generally when people say vitamin A, they mean this specific molecule, Alltrans retinol, and notice the all rather than the al over here. Okay, so we'll start off by having a look at the structure of Alltrans retinol, then we'll go on to Alltrans retinal, and then finally we'll have a look at the structure of 11-cis retinal. I assure you that it's worth doing it this way to give you the broader picture. So, let me start by drawing out the structure of Alltrans retinol. So I'm going to draw this as a skeletal structure, and remember from chemistry, skeletal structures, you don't show carbon atoms, they're implicitly shown by corners. You also don't show hydrogen atoms coming off carbon atoms. Whenever you've got a missing bond coming off a carbon atom, the implicit assumption, the implicit understanding, if you like, is that that will be to a hydrogen atom. So here we go, here is the skeletal structure of Alltrans retinol. So you have a 6-membered carbon ring at one end of the molecule here, and you're going to have one double bond in this 6-membered carbon ring. You're also going to have 2 methyl groups coming off up here, and another methyl group coming off down here. Then you have a very long side chain coming off here, like so. So here we go. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and then you have an alcohol group coming off that final one there, like so. In addition, on this side chain you have quite a few double bonds. It's specifically 4 double bonds. You have a double bond there, a double bond there, a double bond there, and a double bond there. And you also have some methyl groups coming off it. So 1 methyl group coming off there, and 1 methyl group coming off there. So I believe that now is the finished skeletal structure of Alltrans retinol. So to help you understand this molecule in a little bit more detail, let's just discuss the numbering of the carbons here. So the way that we number the carbons is we start off with this one up here, of this 6-membered carbon ring, and we call that carbon number 1. Then we just go round, so we call this one number 2, we call this one then number 3, this one number 4, this one number 5. We miss off these side chains. Then we go to this one's number 6, and then we go along this major chain here. So 7, 8, 9, 10, and I'm just going to dot all of the ones that we've gone over now. So all the ones that I'm colouring in green is the pattern that we're following at the moment. 7, 8, 9, 10. Then we're going to go up to this one, 11, 12, and notice how we're missing out the side chains. 13, whoops, gone a little bit ahead of myself there, 14, and then 15 here. Okay, so we've coloured in, we've named all of the carbons that I have now dotted as green. Now we just need to go back and name the ones that are side chains off. So we go back and start up here, and we name these two, which I'm colouring in now in blue. So this is 16, this is 17, this one down here next on carbon 5, we call that number 18, and then you can work it out for yourself. We will call this one number 19, and this one number 20. So overall, Alltrans retinol has 20 carbons in it. Now, why is it called Alltrans retinol? Well, the reason it's called Alltrans retinol is because of these 4 double bonds in this side chain here. So we've got this long side chain here, and you can see that we've got 4 double bonds there. Now remember from chemistry that double bonds, whenever you have a double bond between 2 carbon atoms, it leads often to there being 2 stereoisomers, a cis isomer and a trans isomer. So the major chains coming off each of the carbons can either be on the same side of the double bond, or they can be in opposite sides. So think about this. These 2 carbons are linked by a double bond. Off each of them, so if we look at carbon number 8 for instance, it has this major chain coming off it, and then it will also have a hydrogen coming off it. Off carbon 7, you've got this major portion coming off, and then you'll also have a hydrogen coming off. Can you see the way I've drawn it is these are coming off on opposite sides of the double bond. This chain is pointing this way, and this major chain is pointing this way. They're on the opposite side. So when you have the 2 major chains coming off the 2 carbons on opposite sides, you call that the trans conformation. Trans means on the opposite side, or opposite, and when they're on the same side, so if they were on the same side, if this was now on this side of the bond, we will call that the cis isomer, and they're not interchangeable without performing a chemical reaction. Okay, so all of those double bonds then, they are in the trans isomer form rather than the cis isomer form. Hence why we call this Alltrans retinol. And the ol bit, retinol, refers to this alcohol group that we've got here. When we turn this into retinal, what we're going to do is we're going to turn that alcohol group into an aldehyde group. We're going to oxidize it and turn it into a aldehyde group. Remember, alcohol groups can be turned into aldehydes, which can be turned into carboxylic acids in the process of oxidation. Right, so that's Alltrans retinol, vitamin A. Let me now show you Alltrans retinal, and hopefully you should be able to work this out for yourself from what I've just told you. So it's a very small change to turn it into Alltrans retinal. So the Alltrans bit tells us that all of those double bonds are going to remain in the trans conformation, and all we need to change is the alcohol group coming off carbon 15, and we need to turn that into an aldehyde group. So this is just nice revision now. We just have to draw the whole thing out again and make a tiny little modification at one end. So here's the 6-membered carbon ring. Of carbon 1 we've got the 2 methyl groups, carbon 16 and carbon 17. Of carbon 5 here we've got carbon 18 coming, and then we've got the proper side chain here, the big side chain, like so. So here are those 3 double bonds, so we're here, then we've got another one there, our final double bond. Then we're up to carbon 15 here, and now we need an aldehyde group off here. So now what's going to happen is this carbon is going to be double bound to an oxygen here, and instead of having this carbon with 2 hydrogens and an alcohol group coming off it, it's now got a double bond to an oxygen, and it will have a single hydrogen coming off, but of course we won't show that. And then we just need to make sure that we put in the correct methyl group, so 1 off carbon 9 there, and 1 off carbon 13 there. So I believe that now is the correct structure of Alltrans retinal. So we're close now to 11 cis retinal. So hopefully from what I've told you, you should now be able to work out what 11 cis retinal is going to be. And what this is telling us is that one of those double bonds is now going to be in the cis conformation rather than the trans conformation. So they're not all going to be in the trans conformation anymore. Some of them are going to be in the cis conformation, in particular the one associated with the 11th carbon, which is of course this carbon here. So we just need to turn this double bond here into the cis conformation rather than the trans conformation. So we need to bring all of this portion of the molecule, which I'm circling now in pink, and we need to bring it onto this side, coming off down here, rather than going off up there. So we need to make a different stereoisomer of this, a different cis-trans isomer of this molecule. Right, so let me now draw this out. So all of this bit's going to be unchanged. So again, drawing out the 6-membered carbon ring here, like so. Here is our double bonds, the 2 methyl groups coming off carbon 1, the methyl group coming off carbon 5. Now we need to be careful. So here's carbon 7, double bond to carbon 8. Here is carbon 9, double bond to carbon 10. There's the methyl group. Here's carbon 11 and carbon 12. And now what we want is this bit down here to come off like this instead. So here's going to be carbon 13. Okay, we'll have the methyl group coming off carbon 13, of course, and we'll have that coming off down here. Then what we'll have is the double bond between carbon 13 and carbon 14, and we need to make sure it's still in the trans conformation, so we need this coming off like here down to carbon 15, and then we'll just have the aldehyde group coming off here. So I hope it makes sense what I have drawn here. This is now in the cis conformation, so this is carbon 12 here. Here's carbon 13 with its methyl group coming off. We then just need to make sure we show the double bond between carbon 13 and carbon 14 in the trans conformation, and it still is in the trans conformation because this is the major group and this is the major group, and they're on opposite sides of the double bond. So this here is how people would usually draw the structure then of 11 cis retinal. Okay, so this is the molecule that you bind to the opsin protein in order to make a rhodopsin protein. Right, so I hope that's now clear that inside these membranes of the discs we're going to have loads of rhodopsin receptors, and these rhodopsin receptors are formed by putting in loads of opsin Gprotein coupled receptors and binding them with this ligand molecule, which is 11 cis retinal. Now, in response to light, when photons of certain wavelengths hit 11 cis retinal, it will change, it will photoisomerize back into all trans retinal. So it changes from this form to this form, so it can absorb photons, and there are photons of specific wavelengths that it likes to absorb, and I'll just go over that now. So rhodopsin, it has specific wavelength photons that it absorbs best. It can absorb photons of multiple different wavelengths, but there are certain wavelengths that it likes to absorb best within the visible spectrum. So I'll draw you a little absorption spectrum graph here. So here are the different wavelengths on the x-axis, and I'll just make sure this is in view. So 400, 500, 600, and then up to 700 here. And what I have on the y-axis here is how well rhodopsin will absorb these photons, specifically how well the 11 cis retinal molecule bound to the opsin protein is going to absorb the photons. So if I just go back up to this picture, when photons come in and hit the rhodopsin, they can be absorbed by the 11 cis retinal. Sometimes, not every photon will be absorbed, but often certain photons will be absorbed, and it will use the energy to transform from 11 cis retinal back into all trans retinal, and this process is called photoisomerization. It's changing the type of stereoisomer that it's actually existing as. Okay, the molecule isn't changing, how can I say, it's not changing the order in which atoms are connected. Okay, so there isn't like bonds, well, it's difficult to know what to say. It's not actually it's changing the isomer of the molecule that it is. It's not actually changing into a thoroughly different type of molecule, but it is changing into a different isomer of this molecule, which is fundamentally a different molecule which will interact with other molecules in different ways. So because the isomerization is triggered by photons, this is called photoisomerization. So 11-cis retinol can absorb photons, and it can use that energy to transform back into Alltrans retinal. And I am currently drawing a graph of which photons it will best absorb. So if you like, you can imagine this is representing the probability that the 11-cis retinal will actually absorb the photon when that photon comes close to it. So it absorbs best at the wavelength of around 500 nanometers, which is sort of in the bluey green portion of the spectrum. It's greeny but still bluey sort of part. Okay, so the graph overall looks something like this. So it's peaking at 500 nanometers. So if you've got a photon of wavelength 500 nanometers, that's the highest probability of being absorbed by the 11-cis retinal, and then it will gradually sort of go down as we go into the sort of reddy portion of the spectrum. Okay, so this is the sort of wavelength then that Rhodopsin will absorb photons best. Right, so you'll get this photoisomerization in response to photons being absorbed, but I want to stress with this graph that photons of wavelength 600 nanometers, so more into the orangey portion of the visible electromagnetic spectrum, they can still activate rhodopsin. It's just that they have a much lower probability of doing so than photons in the green portion of the spectrum. So you might need a lot more orange photons in order to get the same sort of number of rhodopsin receptors actually activated than you would need green photons, is my message with this. Okay, so I hope this graph hasn't confused people too much. Right, so onwards with the story, and this is where it just becomes molecule hits molecule hits molecule hits molecule, which is the nice simple part of the story. Okay, a linear pathway. So we have the photon hitting our rhodopsin receptor, being absorbed by the 11-cis retinal, and the 11-cis retinal is photoisomerizing to become Alltrans retinal. You now have Alltrans retinal bound to opsin. What that triggers is a change in the conformation of the opsin. So opsin with 11-cis retinal bound to it has a different shape, a different topology to opsin with Alltrans retinal bound to it. So once you've now got the opsin with Alltrans retinal bound to it, it's called something different. It's no longer called rhodopsin. Instead, it's now called meta-rhodopsin 2, which is a fantastic name. I don't know who managed to come up with this. So meta-rhodopsin 2. So the rhodopsin is going to change into meta-rhodopsin 2, and I'll stress this is the name for opsin bound to Alltrans retinal. So rhodopsin is opsin bound to 11-cis retinal, meta-rhodopsin 2 is opsin bound to Alltrans retinal, and the major difference is that this molecule will interact with signaling proteins inside the cell in a different way to how rhodopsin would interact with them. So the opsin has changed its structure. So I'll draw this here. So here again, this is the phospholipid bilayer of one of the discs here. Here is our opsin Gprotein coupled receptor. Okay, and now it's got it's not got 11-cis retinal bound to it anymore. It's got Alltrans retinal bound to it, which I will represent in yellow here. So once you've got the Alltrans retinal bound to it, it causes conformational change inside the opsin protein, and now the opsin protein is going to interact with proteins inside the cytoplasm of the cell in a different way. So this molecule here is now Alltrans retinal. Okay, and this meta-rhodopsin 2 is going to activate signaling proteins inside the cytoplasm of the cell and lead to a signaling cascade that will trigger the electrical signal within the photoreceptor. And for short, meta-rhodopsin 2 is often abbreviated down to M2 in that way. Okay, so I think we'll have a break here, and in the next video what we will do is discuss the signalling pathway downstream of meta-rhodopsin 2. So we'll discuss transducin and phosphodiesterase 6 and the cyclic nucleotide gated cation channels that give rise to the dark current. And we'll see how this is actually going to lead to an electrical signal within the photoreceptor. So see you in the next video.